COVID-19 is a single-stranded RNA-enveloped virus that encodes approximately 9,860 amino acids. Gene fragments express structural proteins such as the S, E, M, and N gene, and non-structural proteins such as the 3-chymotrypsin-like protease, papain-like protease, and RNA-dependent RNA polymerase that are encoded by the ORF region. The S protein on the surface of the virus is the key factor that is involved with this infection, as it mediates receptor recognition, cell attachment, and fusion during viral infection. The S protein consists of an extracellular N-terminus and a transmembrane domain anchored in the viral membrane and a short intracellular C-terminal segment. S normally exist in a metastable prefusion conformation. Once the virus interacts with the host cell, structural rearrangement of the S protein occurs, which allows the virus to fuse with the host cell membrane. The spikes that are coated on the polysaccharide molecules to camouflage them invade surveillance of the host immune system during entry. A large number of glycosylated S proteins cover the surface of COVID-19 and bind to the host cell receptor, angiotensin converting enzyme, which also is known as ACE2, through the RBD region of the S1 subunit, mediating viral attachment to host cells in the form of a trimer. The S1 domain contains the RBD, which is mainly responsible for binding of the virus to the receptor, while the S2 subunit is composed successfully of a FP, which is a short segment which is around 15 to 20 conserved amino acids of the viral family, composed of mainly hydrophobic residues such as glycine or alanine, which anchor to the target membrane when the S protein adopts the pre-hairpin conformation, and HR1 and HR2, which form the 6 helical bundle, which is essential for the viral fusion and entry function of the S2 unit. HR1 is located at the C-terminus of a hydrophobic FP, and H2 is located at the N-terminus of the TM domain. The TM domain is also included in the cytoplasmic domain fusion, is responsible for viral fusion and entry. When the S protein binds to the receptor, TM protease serine 2, or TMPRSS2, a type of 2 deoxyserine protease located on the host cell membrane, promotes virus entry into the cell by activating the S protein. Once that virus enters the cell, the viral RNA is released and polyproteins are translated from the RNA genome, and replication and transcription of the viral RNA genome occurs. Also, what happens is the cleavage and assembly of the replicase transcriptase complex. Cleavage of the COVID-19 S1 and S2 subunits is the basis of this fusion. The S protein is cleaved into 2 parts, the S1 subunit and the S2 subunit, by host proteases, and the subunits exist in a non-covalent form until viral fusion occurs. After cleavage of the S protein, the FP of COVID-19 is exposed and triggers viral fusion. Under the action of some special ligands, the fusion protein undergoes a conformational change and then inserts itself into the host cell membrane. Then HR2 folds back to HR1. The 2 HR domains form a 6 helix structure within an anti-parallel format of the fusion core. The viral membrane is pulled toward the host cell membrane and tightly binds to it, and the 2 membranes fuse. Viral RNA is replicated and structural proteins are synthesized, assembled, and packaged into the host cell after the viral particles are then released and dispersed. Now, how is chemistry important in understanding the way in which this virus infects people? Well, it's simple. Biology and chemistry work hand in hand when studying the field of cell biology, physiology, immunology, and much, much more. And life as we all know it relies heavily on biochemical reactions and processes.